Well, good afternoon, everyone. I feel everyone needs a stretch after such exciting science. It's really been terrific, and I think it's a tremendous credit to our young faculty who actually put this program together. I think they were really able to pigeonhole important needs and important questions that you hear being addressed in very exciting ways. So thanks to them for a fabulous program. I thought what I might do is fill in some of the gaps you've heard also, and it's been a little bit easier because some of the things I've talked about have already been introduced by other speakers, but to tell you how we're applying some of the technologies that you've heard about to try to better diagnose the disease, pulmonary arterial hypertension, associated with autoimmunity, connective tissue disorders, but also in the idiopathic variety, and how we're taking the technology not only to help us diagnostically, but to also give us new insights into pathophysiology. So the basic hypothesis you've heard from many in the room, that there's an abnormal immune response affecting the pulmonary arteries that's common in all forms of pulmonary arterial hypertension. So what I'm going to tell you about is some studies that we've carried out. First, a mass spec proteomic analysis, a collaboration with Bill Robinson, where we identified PAH antigens in immune complexes in the lungs, and these studies suggested a novel PAH pathophysiology that was related to abnormal innate immunity. And then I'm going to show you how innate immunity can be abnormally exacerbated by genetic abnormalities in pulmonary arterial hypertension, and how that, in fact, can lead to impaired adaptive immunity. And we're going to talk a lot about changes in T regulatory cells. Mark mentioned CyTOF. I'll show you how CyTOF can be used to establish the presence and nature of abnormally activated peripheral blood mononuclear cell subsets that reflect altered adaptive immunity. And then finally, I'll show you how these observations might relate to new approaches to PAH therapy. And I'm going to mention a lot of work that was carried out by one postdoctoral fellow, Toshi Sato, who's in the audience, so you can ask her further questions. But I'm also going to mention some of the other postdocs in our lab who also contributed in important ways to these studies. So this study was carried out with Bill Robinson, where we said people are defining autoantibodies. Mark Humbert has done a lot of work defining the autoantibodies that are circulating. The Johns Hopkins group with Anthony Rosen had also extensively studied circulating autoantibodies. But maybe the clue is to the antigens that are being produced in the lung that are the site of autoantibody formation and immune complex deposition directly in the lung and directly in the perivascular area. So what we did was isolate immune complexes from lungs of patients with pulmonary arterial hypertension, and we also isolated immune complexes from donor control lungs. And this project was facilitated by the fact that we belong to a network of investigators and transplant centers that collate tissue at the time of explant of the pulmonary arterial hypertensive lung at the time of transplantation. So tissue is readily available. Ruben Tudor belongs to the network. He analyzes the histopathology. Cells are available from different levels of pulmonary arteries, different cells, and there is proteomic and genomic information, as well as extensive clinical information in all patients in the network that have undergone lung transplantation. So we looked at a number of antigens that were present in the immune complex. This is again comparing a small group of about 4 donor controls or unused lungs, and we used idiopathic pulmonary arterial hypertension explanted lungs at the time of transplant. And you'll see some were ribosomal protein antigens, but one in particular caught our attention. And that was not seen in any of the immune complexes in the controls, but was seen, and this is a heat map showing amount, so was seen in the lungs of patients with pulmonary arterial hypertension in their immune complexes. And that was the protein SAM-HD1. And so we verified that there was indeed an increase in immune complexes for SAM-HD1 in a larger cohort of pulmonary arterial hypertensive lung tissue compared to control lung tissue. So SAMHD1 is an interesting protein. It's an antiviral nuclear protein that suppresses HIV replication, and it's part of an innate immune response. Now, none of our patients had HIV, but they may have had other viruses, and we'll get back to that in a little while. But the other intriguing feature is that a mutation in SAMHD1 that prevents nuclear translocation results in a syndrome called Aicardi-Gouttière syndrome that causes a cerebral vasculopathy and a lupus-like pathophysiology or pathogenesis. So we thought we were onto something that might, in fact, relate the specificity of autoimmunity to the pathophysiology of pulmonary arterial hypertension. But we needed more data. So first, we established that indeed the antigen was representative of increased production of this protein in the lung by simply carrying out Western immunoblot analysis on lung tissue from patients with idiopathic pulmonary arterial hypertension, shown here in dark green, compared to control lungs. And then we localized the source of the SAMHD1 in two ways. First, by immunohistochemistry. So you can see, compared to control lung, where you can see SAMHD1 in some of the inflammatory cells that are situated in the periphery. In the idiopathic pulmonary arterial hypertension lung, there was abundant expression of SAMHD1 in the occluded lesions, shown here, and also in the perivascular region, and that was highly significant. So then we wanted to localize the expression of SAMHD1 more specifically to the vessel wall. And so confocal immunohistochemistry was carried out, or immunofluorescence microscopy. And here in red, you see an antigen that represents von Willebrand factors. So these are endothelial cells. And you can see in the nuclei in green abundant expression of the SAMHD1 protein, shown here. And these are in the occluded lesions. You can see also colocalization of SAMHD1 with cells that express CD11C that are likely dendritic cells. You can see SAMHD1 and CD68 positive cells that are monocyte macrophages, not so much in the T cells. And this was work carried out by J.R. Moonen, a recent postdoctoral addition to our lab. So then, well, how's SAMHD1 upregulated? So we thought, well, maybe it's just a product of an inflammatory response. And we looked at a number of cytokines, and we looked at endothelial cells that did express SAMHD1 in the tissues. And if you expose the endothelial cells to cytokines such as TNF-alpha that have been implicated in pulmonary arterial hypertension, you definitely see an upregulation of the SAMHD1 protein in these cultured normal pulmonary arterial endothelial cells. So we said, well, that's fine, but SAMHD1 is an antiviral protein. So is this giving us a clue that SAMHD1 is an innate immune response to some virus localized in the tissue? But what virus? So we called on a collaborator, Dr. Charles Shu, who has developed the technology to really identify the virome in a variety of tissues. And we expected to see some of the common viruses expressed, but really didn't see very much either in the control or in the patient lungs. But what stood out in the analysis was amplified expression of endogenous retroviruses of the HERV-K family, shown here, as you can see in our patients relative to controls. So very little in the controls in blue and a lot in the patients in yellow. HERV-K, there's HERV in general, and most was related to a species, HERV-K2. So then we took this small cohort of about 4 and 4 and amplified it, and sure enough found that the HERV-K envelope mRNA was highly expressed relative to beta-actin in the lungs of IPH patients compared to controls. So what is HERV-K and what is amplification of a retrovirus doing in PAH lung? So it was intriguing because in zebrafish, it's known that are prone to cancer because of genomic instability. ZERVs, or zebrafish endogenous retroviruses, are in fact amplified. So this could be a consequence of genomic instability because in patients with pulmonary arterial hypertension, as you've heard mentioned several times during this meeting, there is genomic instability associated particularly with loss of BMPR2 that could lead to HERV-K2 amplification. Like other viruses, HERV-K2 could definitely induce cytokines that are implicated in pulmonary arterial hypertension, TNF-alpha, IL-6, and SAM-HD1, which is an antiviral protein, member of the innate immune response. And this occurs in macrophages, endothelial cells, and even in tertiary lymphoid tissue. And then, interestingly, the HERV-K DUTPase is actually a locus, SORS1, of a gene implicated in psoriasis. And that gene, or that locus, the DUTPase, has been purified by Maria Aritza, and it can induce a Th1 and a Th17 response. So she's been actively studying this locus and the HERV-K DUTPase product as an inducer of an aberrant Th17 response in psoriasis, but of course it has implications for other autoimmune diseases. And most intriguingly, in a recent study carried out by Johanna Wysoka at Stanford, HERV-K actually induces an antiviral response in embryonic stem cells. So in embryonic stem cells, there's actually a transient amplification of the HERV-K retrovirus in the human genome to protect the embryonic stem cells, but as they differentiate into different tissues, the HERV-K amplification appears to disappear. So again, at least at the message level. So quite interestingly that it can be amplified and it can be repressed, but the DNA is there. But the message and the protein can become amplified under certain conditions. So maybe what happens in the patients with pulmonary arterial hypertension is that the amplification of the messenger RNA from the DNA never really quiets down, or maybe it's reactivated because of some other consequential inflammatory stimulus. So we looked to see whether or not the DUTPase could, in fact, activate cytokines in our cells, as Maria had shown in hers. And in fact, we took pulmonary arterial endothelial cells, and you could see in increasing concentrations of DUTPase related to HERV-K. There's increased augmentation of IL-6, a cytokine that Mark Humbert has extensively studied. We could show IL-6 also in PBMCs, particularly enriched monocyte fraction, and also TNF-alpha. So multiple cytokines can be induced by this HERV-K DUTPase. And this is a work of H. and Cow in our lab. So where is HERV-K hiding out? Where is it immunolocalized? Again, J.R. Moonen looked carefully at the control tissue. Not very much of anything was seen, but in the IPAH lungs, in the perivascular region, in CD68 positive cells, you can start seeing the expression of the HERV-K envelope protein shown here as these green dots against the red CD68 background. So here we can pick it up here, multiple cells in the perivascular region. So this retrovirus is amplified in monocyte macrophage cells in the perivascular region in the tissues from our patients, but not in the controls. And it can induce then in multiple cells through the DUTPase amplification of cytokines. Interestingly, HERV-Ks have also been implicated in altered autoimmunity and in the activation of B cells, which we've also shown measuring the AICDA antigen. And so Toshi looked to see if, in fact, SAM-HD1 antibodies were localized to tertiary lymph nodes that Mark showed you earlier this morning that are seen in proximity to occluded vessels. So what she did was overlay the SAM-HD1 protein and was certainly able to pick up the antibody in the tertiary lymph nodes shown here. So our paradigm at this point, or our model, is that something leads to the amplification or it never dies down of the HERV-K endogenous retrovirus. The DUTPase is released, and that induces heightened cytokine expression in endothelial cells and inflammatory cells that may not die down. As a consequence of this innate immunity, there's increased expression of SAM-HD1 in these cells as well. And as a consequence of HERV-K amplification, there's activation of B cells in tertiary lymph nodes where we see production of the SAM-HD1 antibodies. Together, they form immune complexes and are associated with the activation of complement that can lead to chronic inflammation that we see as a harbinger of pulmonary arterial hypertension and pulmonary vascular disease. Now, what can fuel the system is actually repressed BMPR2, because we know that if you have reduced BMPR2, you have amplification of cytokines like IL-6, like TNF-alpha, and in addition, as I mentioned before, if you have reduced BMPR2, you can also get the kind of genomic instability that might amplify the HERV-K site in the genome. So I'm going to diverge for a moment and then come back to another mechanism of abnormal innate immunity. So we know that when cells are under fire, when there is tissue damage, there's liberation of elastases from neutrophils that we discussed as one of the first lines of defense. But what we see in patients with pulmonary arterial hypertension, and these are the studies of Roham Zamanian, is that it doesn't matter what the etiology is, from connective tissue disease to congenital heart defects, drugs and toxins, idiopathic portopulmonary hypertension, elastase activity circulating in the plasma or elastase levels of neutrophil elastase are significantly elevated versus control plasma. What you also see is naturally occurring inhibitors like elephant that are usually produced in abundance when there's heightened elastase activity are actually depressed. And that's true again in all forms of pulmonary arterial hypertension. So what you see in conditions of inflammation normally is that the elastase goes up and the inhibitor goes up to try to meet it. But what you see here is a disconnect between very high elastase levels and relatively low elephant levels, suggesting a relative deficit in inhibitory activity. So increased neutrophil elastase has been linked to autoimmunity and diabetes, and in fact, it's led to several trials, including one at UCSF, in which alpha-1 antitrypsin is being given to diabetics, and in fact, pre-type 1 diabetics, and those trials are currently underway. And I'll try and explain the mechanism in the next slide. So you have a neutrophil that comes to the site of injury. The neutrophil produces some elastase, and it's been shown that elastase can actually condition dendritic cells to activate T regulatory cells, to really come in and protect the tissue. But what happens is if there's heightened elastase activity, there can actually be transdifferentiation of these anti-inflammatory Treg cells to a pro-inflammatory Th17 type of cell or Th17 response. The good news is that we may be able to control elastase levels, particularly recognizing the deficit in elephant. You can actually add recombinant elephant now. It's been given in Europe in clinical trials as a single dose effect to suppress PM and elastase, at least to damp it down, to damp down adhesion of inflammatory cells, netosis, cytokine production, autoimmunity. And as a bonus, elephant can also improve BMPR2 function, as we've recently published. And then we're going to discuss, Gracia Roncarlo is going to talk to you a little bit more about Treg therapy, which can also suppress polymorphonuclear cells and monocyte activation and autoimmunity, of course. These are normal T regulatory cells, and Mark Nichols' studies have shown that by an unknown mechanism, when you immunoreconstitute with normal Tregs, you can actually increase BMPR2, so you can protect the vasculature, again, as a bonus effect. So, is there a signature, then, of altered adaptive immunity in pulmonary arterial hypertension that can be detected by cytometry with mass spec? As Mark Davis explained, the CyTOF technique that we're using abundantly at Stanford. And can that be used, in fact, as a biomarker to help us titrate our therapies by showing that we can convert abnormal adaptive immunity into normal adaptive immunity, particularly with respect to the regulatory T cell population? So I won't go over this in great detail because he already did tell you the limits of facts that we've all used abundantly by virtue of the fact that the fluorophores have overlapping spectra, so you're limited in a single cell of detecting a number of antigens. But with mass elements, you can, of course, label 100 different antibodies, which have very narrow spectra of overlap. And so you can interrogate by mass spec at a single cell level the cell surface markers, so you can identify where that cell is in the hematopoietic lineage. You can identify receptors for cytokines. You can identify signaling molecules. Once you permeabilize the cells and incubate the antibodies, you can detect transcription factors. So virtually in any PBMC, and it's also being applied to tissue now using the MIBI technology, you can interrogate virtually any antigen in any cell at the single cell level, and you can in fact, as Bill showed you, by barcoding the cells, actually throw a whole bunch of reactions together so you can identify not only what the cell is, but what particular source of stimulation that cell came from in terms of it having been barcoded in a particular well. So in the first cohort of experiments that we carried out, we used a variety of different cytokines. We used IL-6, we used GM-CSF, we used interferon gamma to try to see if we could tweak the PBMCs in patients versus controls to try to establish whether we could elicit a signature that was abnormal. And we also looked at baseline. But actually, it was the baseline that gave us the most intriguing information, probably because these PBMCs are already bathed in a very high cytokine environment. So just to show you how we do some of the analyses, one of them is Spade. And what Spade allows you to do is align cells so that like stays with like. So what happens is cells that have cell surface antigens that are more similar align, and as they're picking up different cell surface antigens, they diverge, and that's how we can build up these hematopoietic lineage trees and identify specific bubbles of particular PBMC subsets. So here's a Spade analysis, and I hope it shows well in the room, which is rather dark, where we've interrogated cells from controls and cells from patients with pulmonary arterial hypertension. This is a representative Spade analysis from a single control and a single IPH patients' PBMCs, but I'm going to show you some data in which we've looked at populations. And in this particular case, we now have the trees built based on cell surface antigens, so NK cells, Tregs, here are the CD4s, here are B cells out here, here are dendritic cells, monocytes, CD8 cells here. So they're aligned close to the antigens that are similar among the cells, and the bubble size tells you how big the population is that is similar to each other. So the color tells you whether or not a particular molecule being interrogated, in this case CXCR4, which is the Sdf-1 receptor, and a number of people have shown you so far that Sdf-1 is actually increased in the tissues of patients, lung tissues in the perivascular region, and in the vessel of patients with pulmonary arterial hypertension. So actually, Kurt alluded to it as well as Mark. So this is the receptor for Sdf-1. And you can see that there's very little activity, so very little yellow or red, maybe a little bit in B cells in the control PBMCs. But in the PBMCs from patients with pulmonary arterial hypertension, you start seeing increased expression of CXCR4 in B cells, shown here that light up in red compared to the B cells here. Regulatory T cells are in here, not too much, but the T cell population in general, the CD4 T cells are a little bit brighter, green compared to blue. And look at the population here, the classical dendritic cells at the bottom are red compared to greenish and orange. So we can start picking up at the individual cell level particular markers of activation that are relevant to disease pathology and others that we didn't know were relevant. So this was a good guess based on knowing that Sdf-1 was increased. It could explain how some of these cells can get recruited to the lung, particular to the perivascular region that expresses high levels of Sdf-1, and actually sit there and maybe organize as tertiary lymphoid tissues because all the elements are there, B cells, T cells, dendritic cells that can be called in. Okay. This is very interesting because this was a signature that I'm showing you in a single PBMC isolate from a patient with pulmonary arterial hypertension versus controls, but as I'll show you in a moment, it was representative of all the patients with pulmonary arterial hypertension that we studied. And that's heightened activity of pStat1 in CD4 plus CD25 high cells that include the regulatory T cell subpopulation. So we're going to look at these cells here and here. Okay, so they're green in the control cohort, and they're yellow and red and bright green in the patient cohort. So we collate data from numerous patients, numerous controls, and then we analyze it together. So this represents 12 controls that are carefully phenotyped as part of a study being carried out by Francois Haddad, looking at the immune system in aging and correlating cardiovascular function with immunology. So we have a lot of good phenotypic information to confirm that they're normal, don't have any pulmonary arterial hypertension or left ventricular dysfunction. These are about 10 patients with idiopathic pulmonary arterial hypertension, PBMCs, and you can see there's elevation in pStat1 and circulating CD4, CD25 high T cells. Same is true for scleroderma, same is true for drug and toxin PAH, and there are about 10 here and 8 here. So a signature that seems to be representative of all the subsets of pulmonary arterial hypertension patients. The pStat1 activity in this T cell subpopulation that we're now further refining to determine what the nature of the regulatory T cell really is in this CD25 high subpopulation. So transdifferentiation of regulatory T cells to pro-inflammatory STAT1 positive cells has been implicated in rheumatoid arthritis in 2 recent papers. So what we think is going on is what I alluded to before, that you have a T regulatory cell that is no longer protective, but is actually a cell that could possibly contribute to the disease if it gets there. So then the question is, does it get there? And as I mentioned, we looked at the GM-CSF receptor alpha because we had shown that there was high expression of GM-CSF in IPH tissues, as I'll show you in a moment. And in fact, this subpopulation of cells does have, in at least 2 of the 3, and maybe this is verging on significance, the scleroderma subpopulation, high levels of receptor for GM-CSF in this subpopulation. So that means that these pStat1 positive cells that are abnormal, seen in the patients with pulmonary arterial hypertension, could in fact be recruited to high GM-CSF locations, which we've shown are the pulmonary arteries in patients with pulmonary arterial hypertension. So this was work that we carried out a number of years ago, where we looked at expression of GM-CSF, which Bruce has told you about, in controls in response to TNF-alpha and in patients with BMPR2 mutations and found it to be elevated. And then what we did was knock down in endothelial cells BMPR2 with siRNA, stimulate the cells with TNF-alpha, and show that there was a 2-fold increase in the production of GM-CSF when BMPR2 was deficient. Now, this was interesting because it wasn't on the basis of increased transcription of GM-CSF, but on the basis of increased mRNA translation. And when cells are under fire, they undergo a protective ER stress response that involves production of stress granules that keep mRNAs from being very actively translated, or at least mitigate the response. And what happens here is that there's abnormal translation of cytokines, so not only GM-CSF, but also IL-6 and IL-8 were documented. Work of Hiro Sawada. And this just shows you high expression of lung GM-CSF in patients with idiopathic pulmonary arterial hypertension compared to controls. This was in his original publication. And we look, as Bruce mentioned, very little expression by immunohistochemistry of GM-CSF in the lung normally, particularly with reference to the pulmonary arteries, but in pulmonary arteries with lesions shown here, very high expressions in the endothelium, the intima, and also in the media and in the perivascular region as well. So this suggested, in fact, that you have the ligand that can actively recruit cells that express the receptor for GM-CSF, among them cells such as monocytes, macrophages, but also these abnormal T cells that can actually contribute to the inflammation. So in view of this, there are promising therapies on the horizon that can actually control aberrant immunity, aberrant innate immunity that leads to aberrant adaptive immunity. And one of them is tacrolimus in low doses, activates BMPR2 and is an immunosuppressive. And what Ed has referred to and actually brought to our attention is a recent article in which low-dose tacrolimus actually increases the function or improves the function of regulatory T cells. So there's promise in using low-dose tacrolimus, which she's shown is highly effective in preclinical studies and compassionate use in the treatment of patients with pulmonary arterial hypertension that exhibit these abnormalities in innate and adaptive immunity. And the second treatment that we are bringing to clinical trial, hopefully with at least phase 1 in the next year, is Elephant, which is a recombinant protein that has been given subcutaneously in preclinical studies that, like low-dose tacrolimus, can reverse extensive pulmonary vascular pathology in experimental animals. It can also reverse or regress intimal lesions in organ culture from tissue with patients with pulmonary arterial hypertension. And it also activates endothelial function by increasing the function and the signaling capacity of the BMPR2 receptor. And you've heard of other treatments like bistatin, the leukotriene B4 inhibitor, IL-6 receptor antagonist, T-regulatory immunotherapy. You're going to hear a lot more from Maria in just a moment. So in summary, what I've shown you is that there is an amplification of an endogenous retrovirus that occurs in pulmonary arterial hypertension and that can induce a chronic, innate, immune antiviral response that doesn't go away, that can lead to the deposition of SAM-HD1 immune complexes. Heightened elastase activity is another aberrant innate immune response in pulmonary arterial hypertension. It can lead to abnormal adaptive immunity and autoimmunity via Treg transdifferentiation from a protective cell to a pro-inflammatory cell. Cytoph analysis reveals a pro-inflammatory subpopulation of PBMCs that can include these aberrant T-regulatory cells. And loss of BMPR2 amplifies GM-CSF recruitment of pro-inflammatory GM-CSF receptor positive cells, including these abnormal Tregs. So here you have revved up of an abnormal GM-CSF process. But promising PH therapies are being introduced that address abnormal innate and adaptive immunity and that are highly efficacious in preclinical studies. So I'd like to just end by thanking the people that I've mentioned that have contributed to this work, other folks in our lab that supported the projects that I've described to you, and a variety of wonderful collaborators, Gary Nolan and Wendy Fantel for the Cytoph with their postdoctoral fellows, Bill Robinson and his Mark and Roham, Richard Bland, Joanna Wasoka, Charles Shu at UCSF, Oliver Wedo at Kiel for work with Elephant, and Maria at Ohio State University. Thanks very much.
All right, I think we can take one question.
First of all, that was a really wonderful talk. The question I have, do you have any idea if indeed HRVK skew the cells towards Th17 phenotype because downregulates FOXP3? The virus, the retrovirus that you think it's implicated. You know, some of these retroviruses indeed, in addition to providing an inflammatory environment, can downregulate FOXP3 and really skew the cells towards Th17 phenotype.
Right. So one of the things we're doing now, of course, for whatever reason, FOXP3 wasn't included in our initial CyTOF analysis. It is going to be included in our analysis now with a variety of other regulatory T-cell markers that you and others have described to be important in the immune regulation. So we should be able to answer that, and we should be able to correlate that subpopulation with expression of HERV-K, or we could even test regulatory T cells following exposure to DUTPAs to see if FOXP3 is downregulated. That would be a good experiment to do based on your suggestion. Yeah. Ruben? Yeah.
Do you have any evidence that the retrovirus is present in BMPR2 heterozygous without disease in the peripheral blood?
We haven't looked at that, but we can, and we could even look at it in the, and in fact, interestingly, Joe Lascalzo asked me the same question. He said, have you looked at your iPSCs to see if it's amplified in those cells? Because we have not only the BMPR2 patients, but we also have the carriers that don't have the disease. So it would be very interesting to know if there's a difference there.
What's related? This immunocomplex also increase in the, the carriers without disease? The immunocomplex.
Oh, we haven't, you mean circulating immune complexes? We haven't looked, in fact, we don't detect them in the circulation, we only detect them in the lung. That would be the limitation there.